Hi guys, how are you all doing? I hope you are doing good in this corona situation. This is a 60 days and 60 topics class system, and this is a lecture room which is created by me, and this is very helpful for CSIR, ICMR, DBT, JRF, and NET exam. So I am Ashish Gupta, and I am going to discuss this topic today. And as you all know, I am the owner of the Medical Acme India channel. Please do subscribe to my channel if you have not subscribed yet. So what about this 60-day plan? 60-day plan is very helpful, and this is very in a arranged pattern. You will see that later there is a day 1 to day 60 plan for you, and if you are doing yourself, then it is also good for you. This is a 2-month strategy to crack JRF exam, and this is very important. This is very kind of a very scheduled program, you can say, and this is guaranteed 100 marks in CSIR JRF exam. So 60 days are enough to cover the 60 important topics of the life sciences, CSIR life sciences. So what happens, there are many topics. There is a very vast syllabus in the CSIR JRF, but what happens, there are selected topics which are repeatedly asked in the exam. So these repeatedly asked topics I have gathered from the students and taken review from many students, and then created a program of 60 days for 60 important repeatedly asked topic. And these topics are very important, and this can be covered in 60 days, and how it can be covered and how it is covered you will see later. But what happens, you have to give at least 2 hours of study in a day. So this 2 hours will lead to you like 120 hours of study, and 120 hours of study is a very good thing for clearing the CSIR, ICMR, or DBT, JRF, or any kind of entrance examination of JRF. JRF is not a tough exam, but it needs a regular and scheduled plan for the preparation. So preparation for this exam is very important. So I am going to help you for preparing the examination. If you are having any doubt in reference books, please check for link in description box. You will get the reference books for different topics. Appropriate plan for December 2020 exam. No one knows when is going to happen the December 2020 exam, but perhaps it will be in the month of March. So for March you are having 60 days, proper 60 days, and here you'll get a proper 60-day plan for your study. So what happens, here is you will get a day wise, means day 1 to day 60 while splitting off for your convenience of preparation for the CSIR JRF. So let's start. So this is my channel Medical Acme India. Please subscribe to my channel, and please share my video to everyone your friend, and this is exclusively for the Medical Acme India viewers. So at day 0 you are going to study the fundamentals of biochemistry part, like amino acids and genetic codons and structural organization of proteins on the day 2. For these 2 days you are going to study about the amino acid and proteins, the structure, solubility, all these things you will cover in 2 days for the fundamentals of biochemistry part and basically the protein part. So after having this is, you will see that the advancement in the topic, the complexity of the topics are gradually increasing. First I'll start with the simple topics, and then I'll go to the advanced or complex topics. So this will be helpful that you're the portion which is getting vast in the complex topics is clear in all the previous topics. Like if you don't know the amino acid, you are not going to have any idea about the translation of the mRNA. So it is very important to study amino acid first. And the second part which becomes important is the cell bio. Cell bio is very important for the examination point of view. Any exam you can say, DBT JRF, ICMR JRF, or CSIR JRF. These 5 topics from the cell bio and cell signaling is very important. You can cover it in 5 days. Cell cycle, regulation, CDK, and all the CDKs and all the CDCs, they are important. So you must have a knowledge about all the complex, all the signaling, all the cycle regulation, cell cycle regulation part, and all the checkpoints should be clear. What are the CDKs which are used in the which part of the checkpoints. So what happens ki cell, after that you will have to study in the day 4 the cell signaling, day 5 will the cell junction, day 6 for mitochondria, basically focusing on the mitochondrial matrix and NADPH transport, NADPH system, and etc. The etc means basic part of the etc. You will later study that, you are going to study later the etc as a full system, but here you must have to clear about the basics of the mitochondrial matrix, Christie, and components of the inner and outer mitochondrial membrane. For day 7, apoptosis and necrosis, that is cell death, you are going to study on the day 7. For day 8, you have to start the mol bio part, and mol bio part start with the nucleosome and histone, mainly the histone modification system, histone modifications where the histones are modified, and what are the euchromatin and heterochromatin part of the histones, with how they are created. So for day 9, you are going to study about the structure of RNA, pDNA, mRNA, siRNA, and reverse transcriptase. All these structures must be clear. Sometimes what happens, they ask the structure based questions from these miRNA and siRNA silencing based questions are also asked. So must read these 5 structure and their and their benefits in the mol bio system. So what happens that day 10 you are going to study DNA repair mechanism, all the 5 nucleotide excision, direct excision, all the 5 excision systems are very important. Nuclear DNA repair mechanism system is very important, so you must go through all these. NHEG is also very important, must go through all the 5 DNA repair mechanism. For day 11 you are going to study telomerase activity. Telomerase nowadays are in very hotspot for the question asked in the CSIR JRF or the DBT JRF. So transposons, as you have seen that ki 2020 wale exam mein they have asked transposons in both the CSIR exam and in the DBT exam, so it becomes very important. So must go through transposons on the day 12, and from day 13 to day 20 you are going to focus yourself in the bacterial genetics or recombinant DNA technology. All the bacterial or bacteriology wala part you are going to study from the day 13 to day 20. From day 13 you must start with the antibiotic action, like what are the antibiotics and where they are going to hinder the bacteria. So then day 14 and 15 will be gene regulation in bacteria, lac-operon, tryptophan operon, and day 16 will be conjugation and HFR strains. This is very important topic. Again I am saying cloning vector and expression system, restriction enzymes, modification system, restriction mapping, and practicing all the numericals based on the restriction mapping is very important. Must go through it. And for day 20 you must start with the techniques like RFLP, RAPD, qPCR, and DNA fingerprinting. You can cover, don't go like in details, but what happens, you allot 20 to 25 minutes for each and find a proper video on the YouTube. You will find, and if you are interested in my lecture room you can join, and I'll give description in the last slide. For day 20, and from day 21 to day 25 you must clear the basic chemistry part, that is equilibrium constant, pH. You have seen that pH say they ask questions, order of reaction sometime asked in the ICMR exam or DBT exam, drug dose response were asked in the DBT exam. And so these things becomes very important. Half-life reaction, radioactive decay was asked in the DBT exam, so it becomes very important. You must clear all these the formula based numerical types of questions. You must go through on day 23 the technique of chromatography, all the chromatography, ion exchange and gas exchange, all becomes very important, and must clear all these chromatography techniques. Day 24 go through the Michaelis-Menten equation, and then the enzyme inhibition, all the 3 kind of inhibition, competitive inhibition, uncompetitive, non-competitive, all kind of inhibition, what are the effect of on Km and Vmax. Must read all these things. And then on the day 25 must you read the proton pump, sodium pump, all the pump, how these pumps are works, and what are the glucose transporters which are used in intestine, brain, and all these must be clear to you all. So what happens from day 26, then you are going to start the plant physiology part, that or biochemistry part. Here you are going to study for the Kelvin cycle, that is C3, CAM pathway, and C4 and C2. These two cycle you are going to study on day 27 and day 28 with the electron transport chain. Here you are going to study the glycolysis and Krebs cycle, ketogenesis, HMP shunt, that is pentose phosphate pathway, gluconeogenesis, urea cycle. These are the basic, these are the metabolic chemistry part, or the you can say ki plant physiology part. So you must go from day 26 to 34 all the plant physiology or metabolic biochemistry part. Then you are going to start with the botany. Two important botany topics are here for the phytohormones and its signaling, and photoperiodism and vernalization. Actually, this time plan is for the zoology or life science people, because they don't read much botany. So what happens, these two topics are only for the botany. Otherwise, Kelvin cycle to gluconeogenesis, these are also the botany part. So day 37 is for the animal physiology, and you start with the muscle contraction, action potential on the day 37, day 38 with the regulation of respiration, and day 39 for the excretion and counter current mechanism, that is absorption kind of thing in the nephron. And on the day 40, the last day of physiology, you are going to study about the electrocardiogram and oxygen dissociation curve, chloride shift, and Hamburger's effect. So all these things, now you are up to here, you are clear with all the physiology part. Then what happens, you start with the developmental biology. Developmental biology is only three topics which are important, as cleavage, neurulation, and gastrulation, that is epiboly, involution, invagination. All these terminologies must be clear in all the stages of the development. So induction and competence becomes very important, so must focus on the genes which are involved in the induction and competence. And for axis formation in Drosophila, you must study on the day 43, and it is very important for axis formation or pattern formation in what Drosophila. Many genes are involved, like nanos and hunchback. These are very important, WNT. So must read all these genes in detail, all these genes in details. From day 44 to day 51, you are going to study about the ecology or genetics part. Ecology and genetics, they both comes under the experimental biology part. So must read these things. Speciation say you can start, and then you can study the population dynamic, Heisenberg principle. Must solve the must solve numericals here. From genetics, there are many numericals which are very easy and asked, and people, what happens ki they leave the numerical because they think that this is very complex, but they are actually very simple. For epistasis on day 46 from and then linkage mapping, you have to solve the numericals. Then go for Drosophila mutants like eye, wings, and all mutants of the Drosophila, and then must solve the pedigree analysis questions. For then on day 50, you are going to study about the somatic hybridization, must clear about the HGPRT medium wala part. And then on day 51, you just end up the genetics and ecology part with the tetrad analysis. Now you have two topics from the ecology and ethology, that is animal behavior, altruism and kin selection, and Lotka-Volterra curve and species richness. Don't go in detail, but you must have a brief idea about what are the topics, what are the formula used in the species area richness. So on day 53, you are going to start with the immunology part. Now come to the immunology, last part of the lecture room on JRF 60. So from day 53, you are going to study the TLR and TLRs and other receptors of the immune receptors. So for on day 54, you are going to study about antibodies and their gene arrangements, and then go ahead with the immunization and vaccination, MHCs, endogenous and exogenous pathways for antigen presentation, B cell and T cell differentiation and activation, and lastly the ELISA, FRET and FRAP-like techniques. Then comes with the very advanced kind of topic, that is screening of transformants, blue-white selection, and the microarray. These are the screening of the transformant kind of thing, is a very advanced type of, or you can say the higher level of topics. So must cover this these topics, 60 topics which are very important for the examination point of view. I hope you have No doubt, please take a screenshot of all these slides which I have discussed, and you will have a very good benefit. Please subscribe to my channel. This is a humble request to you all. And if you want to join our JRF-60 lecture room, you can inbox me on Telegram. It is one-time payment of 999, and the room will open on 1st of January 2021, and the lecture will go like from day 1 to day 60, from 1st January to 1st March. And once again, I would like to tell you that this is a very delicate and very essential kind of lecture room. You will get definitely benefited from it. There are many different YouTube videos and the videos of some paid subscriptions which I have planned, and I have took in the our lecture room for JRF-60. Please do make an effort and contact me. You can message me on Telegram @Ashish Gupta is my username. Thank you so much.